Great. Okay. So the point of this is that you can literally look at the biochemistry at every place along the way. You can look at what happens to cellular signaling, what happens to synaptogenesis, what happens to your ability to fight infections, what happens to your likelihood of developing a thrombosis, to your inflammatory status, to your neurotrophin signaling, to your neurotransmission. It's really amazing. And so when you are in this connection mode, which is where you're now cleaving your APP at a single site, you're making SAPP alpha and alpha CTF, which are both one outside the cell, one inside the cell, are literally telling your neurons make and keep synapses. So you are maintaining and producing memories and things like that. And on the other hand, this same molecule, APP, when it recognizes that there are pathogens, when there is low estradiol, interestingly, when there is any of these changes that we always talk about, inflammation, toxicity, heavy metals, things like that, it will shift into this alternative mode where you now cut at three different sites. NF-kappa B, by the way, is one of the things well characterized that causes this shift. So anything from inflammation. And so now you're making four peptides, SAPP beta outside the cell, amyloid beta, which actually breaches the membrane. So you have some of it outside the cell and some of it inside the cell. And of course, that's the one that's been vilified in Alzheimer's. It is part of your immune system response. It's part of your body shifting into this protection mode when you get COVID, when you have low blood flow, when you have sleep apnea. All these things will shift you toward that protection mode. The third peptide is called JCASP because it's juxtamembrane. It's just inside the membrane. And then the last one is called C31, and actually one that we first discovered and described in the lab years ago. And this is the 31 amino acids at the C terminus of the APP. And we found and published that it is another cytotoxic peptide like A beta. So these things are telling your body, I'm switching over to protection. I'm going to kill the incoming pathogens. And I'm going to shift my resources away from making and keeping synapses. So it's part of switching from connection to protection. Interestingly, the tau changes. So the tau normally is like a bolt. It's bolting your microtubules into place. It's making your neurites nice and supported and strong and allowing you to make and keep this change in behavior. You're now forging new pathways and stabilizing those pathways. This same molecule, when you now have shift into protection mode, gets phosphorylated, pops off the microtubule, and now it is an antimicrobial protein. It's a little bit like, the militias from the Revolutionary War. You can be farmer one moment, then you can be a musketeer the next moment, and that's the same thing. P-tau is a stabilizer, and tau is a stabilizer, and p-tau is antimicrobial, and it is also a prion, so it begets more of itself because it is now ramping up. You're going out and you are engaging and enlisting more of the militia to fight the ongoing problem. And so you can see this all happens coordinately. Everything happens. And interestingly, the p-tau, there are several kinases that phosphorylate it. The most common one and the one that's been studied the most is actually GSK3 beta. So glucose synthase or glycogen synthase kinase 3 beta. And then something called the epithelial mesenchymal transition, which occurs interestingly in cancer and also in fibrosis, also occurs with Alzheimer's disease. And we published that years ago. You see this, and it alters the organization of cells, the respiration of cells, and the energy of cells. You're switching from more of a oxidative phosphorylation state, efficient use of energy. You're now switching into more of a glycolysis state, much again like you see with cancers. It's part of your going into protection mode. Thrombosis. You go through from an antithrombotic mode to a prothrombotic mode by several mechanisms. And so, no surprise with things like COVID and Alzheimer's, you see this increased risk for many microthrombi. Your immune response, you switch from an anti-inflammatory response to a pro-inflammatory response. Tryptophan metabolism, interestingly, you go from making serotonin to making quinolinic acid. So you're now, this is a microglial, antimicrobial inflammatory process. The ApoE signaling, very interesting. So ApoE4 in connection mode is off the DNA, and it prevents the repression of these 1,700 genes that we discovered years and years ago that are inhibited by ApoE4. And so you can actually see, for example, SIRT1 is one of many examples. And so you see this when it's off the DNA, you make your SIRT1. You can see the RNA polymerase bind. You make the SIRT1. When you switch over to protection mode, the ApoE4 is down on the DNA, and now you're preventing the production of SIRT1. You're preventing this mode of connectivity, and you're switching over to this protection mode. And one of the things that you inhibit are these various factors that normally will dampen down your immune response. So you dampen them down, and now your immune response is higher. So we have a very exciting, actually, a molecule that makes your ApoE4, it negates its ApoE4 effect and switches it back so that it acts like ApoE3 and removes its likelihood of its risk for giving you Alzheimer's. Blood-brain barrier switches from tight to leaky. Your GSK3 beta goes from inhibited to active, and then it phosphorylates your tau. And by the way, one of the things that the ApoE4 inhibits is one of the subunits of PP2A, which is the phosphatase that takes the phosphorylation off the tau. So when in the presence of ApoE4, you get more phosphotau. You jump into that protection mode more easily. Your A beta polymerization. You have monomeric non-toxic A beta in your connection mode. When you've got the protection mode, you are switching. You're now oligomerizing your A beta. It is now antimicrobial. It is now a prion, and you can actually inhibit that with homotaurine. In fact, actually, someone needs to come out with a good, clean, and reliable homotaurine, because it's actually an interesting molecule. There's of course, this current study, TB006, which is another way to inhibit the polymerization. This is related to another mechanism that's involved in A beta polymerization. That's actually a pectosol related mechanism. So again, inhibiting that, you're inhibiting this immunological response. Neurite outgrowth. Interestingly, in the connection mode, you have a high level of renin. And what's been shown recently is in people who have high renin, even if they've got a lot of amyloid around, they tend to do well with their cognition, whereas people who are in the low renin state do poorly with their cognition. Well, renin is just like a machete that's chopping. If you're in a jungle, you need someone with a machete to chop this thing down. This is a protease that lives in the extracellular space and allows you then chopping these things, it chops up proteins, allows you to put out those neurites, allows there for better movement. And so no surprise, when you are in protection mode, you have a downregulation of your renin. When you're in a neurite outgrowth mode and a connection mode, you have high renin. And before that, during development, it's actually used for moving the cells through the extracellular matrix. And then there's a pathway called hyposignaling, which is autophagy related. In connection mode, you have autophagy activated. In protection mode, you have reduced autophagy, which allows you to keep these peptides that are antimicrobial around. So you increase your A beta. So what's amazing is that these and many other pathways are all coordinately affected, so that you're literally making this mode switch from one to the other. Now, the reason I show this is because we're all interested in anything that will continue to improve people's outcomes. And this can be that made photobiomodulation, great example. stimulation, magnetic stimulation, great example. Low dose naltrexone, especially for people who have autoantibodies, another great example. So everybody, we're all open to whatever gives the best outcomes. Now, having said that, just today, I know Ann and I and others saw that it's claimed now that there's a new surgical procedure from China that cures Alzheimer's. Well, unfortunately, they haven't published, as far as I could see, they haven't published anything that showed data that show, yes, this improves. So we all want to see data. We want to be convinced. Now, yes, it's it's tough to say we won't believe it until there are, 10 randomized controlled, double-blind, placebo-controlled crossover trials. So at what point do you pick it up and say, look, this looks pretty convincing? I think, you need to see at least, some anecdotes where there's documentation. They're different from someone saying, I feel better, because that can happen from a lot of things. The great news is this thing that you mentioned here, this LifeWave thing, it's very good for making a placebo control because it's a patch. This is a patch that is supposed to reflect the body heat back into the body and induce stem cells. So very, very important for something like this to see data. And especially, it'll be great, again, if you have a patch that's a placebo patch. Take 10 or 20 people and do it with the placebo patch and with the real patch and see and just document their outcomes. Exactly what we did in our earlier trial that Anne was one of the PIs on, and very successful. This is published, freely available online in the Journal of Alzheimer's Disease, followed up by Dr. Heather Sanderson's a trial where she did very much the same thing, got very much the same outcome. So it wasn't just that we did it and nobody else did it. We've got now, confirmatory results. So I would love to see the same thing for this patch that you're mentioning. Please let us know as soon as data become available.